Alright, here today we're going to look at the Calvin cycle. The Calvin cycle right here. So the Calvin cycle we can think of as a reverse Krebs cycle. In the Calvin cycle, so this happens after all the light reactions. We've made our NADPH, we've made our ATP. Now we're going to use them to make glucose from carbon dioxide. So we'll start off with something familiar. In the Krebs cycle, we have our products and our reactants. So we start off with acetyl-CoA. We start off with ADP. We start off with NAD+, and we go through this cycle of oxidizing pulling off electrons to make NADH, FADH2, ATP, and carbon dioxide. We're breaking glucose apart to make store some energy in NADH and FADH2 to use in electron transport chain, making some ATP on the way, byproducts being CO2. In Calvin cycle, we're reducing carbon dioxide. Here, we're oxidizing organic compounds. Here, we're reducing, adding electrons on to carbon dioxide. Our inputs, what are we putting in? CO2. We're putting in ATP. We're putting in NADPH. What are we getting out? Outputs. We're getting out used up ADP, used up NADP+. But most importantly, we're getting out G3P, which will be later on used to make glucose. So we'll think of G3P as glucose. So the inputs are basically the outputs of the products. If you think about it, inputs here are the outputs of the Krebs cycle. Pretty much the exact opposite. The outputs, basically what we put into the Krebs cycle. Alright, so the Krebs cycle produces 6 NADH, 2 FADH, and 2 ATP from catabolically breaking down glucose. It releases energy. The Krebs cycle starts with acetyl-CoA. However, the Calvin cycle requires an input of 6 NADPH and 9 ATP to anabolically build up reduced carbon dioxide into G3P, which is later glucose, and it requires energy. So these are opposites. Alright? Both are cycles, they both regenerate their starting material. So in Krebs, when we put in acetyl-CoA to help take oxaloacetate and turn it into citrate, we go through the cycle and we get oxaloacetate back again. Same thing with the Calvin cycle. We put in CO2, we do that to take RUBP and turn it into 3-glyceraldehyde. Right, and we go through and then we get RUBP back. It's all a cycle. But Krebs cycle, oxidation, catabolic, breaking down, releasing energy. Calvin cycle, anabolic, costing NADPH and ATP to build glucose. This is going to have a positive delta G. This is going to have a negative delta G. So the Kelvin cycle, which I have a picture here in, it's 3 steps. The first step is carbon fixation. Right, we take 3 molecules of carbon in, and this enzyme called ribisco, ribulose bisphosphate carboxylase oxygenase, that's the full name, fixes carbon dioxide and puts it on RuBPH, ribulose bisphosphate, and it breaks apart into 3 molecules of 3-phosphoglycerate, our starting material. Alright? So, carbon fixation, new CO2 incorporated in by ribisco into the cycle, into making our 3-phosphoglycerate. Alright? So carbon fixation, we add carbon to ribulose bisphosphate to make 3-phosphoglycerate. We add carbon dioxide into the chain. The next step is reduction. We use ATP energy and electrical energy stored in NADPH to reduce this 3-phosphoglycerate down to G3P, our glyceraldehyde 3-phosphate. This is what gets turned into glucose. So we reduce this down, we're adding energy to reduce it down into basically glucose. In our third step, we're going to regenerate. We're going to regenerate our RuBP, or ribulose bisphosphate, by using 3-ATP. We're going to use 3-ATP to rephosphorylate 5G3P into RuBP. So, things to note. When we input 3CO2, we're going to get out only 1 G3P. That is 1/2 of a glucose molecule. So for each 3 CO2, we get 1 G3P, 1/2 of a glucose. We need 6 CO2 to make 1 glucose. We need to go around this cycle twice, 2 times to make 1 glucose. So it would take 18 ATP, 12 NADPH to make 1 molecule of glucose. This regeneration phase, we're just remaking the RuBP that we started out with. We input 3 CO2, we get out 1 G3P, or glucose. So, that is basically the Kelvin cycle in a nutshell. But it's been a long evolutionary trek to get there. Plants that go through photosynthesis just like this are called C3 plants. They're called that because they fix CO2 initially to form 3-phosphoglycerate. They initially fix CO2 to make 3-phosphoglycerate, a 3-carbon molecule. These plants include many of the common plants you know, like crops like wheat, rice, grains. So they're all called C3 plants. When it gets really hot outside, so really hot, plants start to lose water, and they need water. They need it to conserve H2O to stay alive. So what they do is close these little holes in their leaves called stomata. And they use stomata to take in carbon dioxide. But they close those off when it gets too hot to lose water. So oxygen starts to build up inside of the plant leaf, and carbon dioxide, since it can't get back through the stomata, starts to be really scarce. What happens because of this is something called photorespiration, where oxygen is actually fixed to RuBPH, RuBP, ribulose bisphosphate, and produces carbon dioxide as a product. But just like respiration, but no sugar is created, and ATP is still used to make this. No nonsense, don't use it for anything. molecule. Photorespiration is bad. It's not good. It costs energy, but it doesn't produce anything we can use like a glucose molecule or ATP. Plants want to limit photorespiration. Not good. And there's some evolutionary mechanisms that they've developed to be able to do that. In specific, there's two other types of photosynthesis besides C3 that plants have developed over time, specifically that live in hot climate, to limit photorespiration. There are some plants called C4 plants. They're called C4 because this first molecule that they fix is actually malate. It's got 4 carbons. C4 plants open their stomata only slightly when it's warm, so they minimize H2O loss, but they can bring in some CO2 that they fix, that they store as malate in the very surface cells of their leaf. They're called mesophyll cells. And they store up that malate in those mesophyll cells, that only these mesophyll cells are losing water, only a little. And then they ship that malate out to the Calvin cycle, the inside of the bundle sheath cells, so farther down in the plant, where that malate can be used to make glucose in the Calvin cycle. So in C4, they separate CO2 fixation from the Calvin cycle. They separate the first phase from the rest of the cycle, and they separate it spatially. The CO2 fixation happens in a different place than the rest of the Calvin cycle to help limit water loss. So organisms like sugarcane are C4 plants. Vascular, very wet or succulent plants like pineapple have another strategy called CAM photosynthesis. CAM photosynthesis, they separate fixation and the Calvin cycle, just like in C4 plants, but they separate it. Chronologically. They separate it by time. So at night, these plants open their stomata, let in all the CO2, and fix that CO2 into malate. Then they close it off during the day. During the day, when it's hot, they close off their stomata, limit water, and then use that malate to do the Calvin cycle. Light reactions are happening during the day. So in CAM photosynthesis, they separate carbon fixation in the Calvin cycle time-wise. In C4 plants, they do it during the day too, but they separate it spatially. Some cells on the outside versus the inside of the leaf. These are adaptations that certain plants have derived to be able to maximize sugar production and decrease water loss. So some ending notes. Sugar produced in photosynthesis can be linked together to form polysaccharides like cellulose. Cellulose is the most abundant organic molecule in a plant and probably in the planet. So, just how like we store energy in glycogen and fat, plants do in cellulose and starch. Cellulose produced in photosynthesis can be used by the non-green parts of the plant, so only green parts of the plant can photosynthesize for energy via cellular respiration. Right? Look at this. This is a very good simple diagram review of what happens in the light reactions and now the Calvin cycle. Things to pay attention to do. The basics of the Calvin cycle from today and C4 versus CAM versus C3. Oh, sorry about that. If you have any questions, please let me know. Otherwise, I'll see you in class.